Hello everyone. In today's video, we will be looking into how a tube of blood can pave the way for decoding and possibly curing the most convoluted and lethal non-infectious disease to date called cancer. Welcome back to my channel, The Curious Biotechnologist. My name is Garima Singh. Please subscribe to my channel if you have not done yet. Peripheral blood can be a great source of cancer-related information due to presence of circulating biomolecules like circulating free nucleic acids released by several cell types, including many cancer cells. These nucleic acids can be either circulating tumor DNA, which could be screened for the presence and expression level of cancer-promoting gene called oncogene, as well as tumor suppressor gene. Other elements in circulation like circulating pre-mRNA and mature mRNA can be very useful in relation to cancer. In addition to the circulating nucleic acids, blood also carry along several cells or cell-like bodies other than immune cells like circulating tumor cells or CTCs, exosomes, tumor-educated platelets that can be useful source of information and play important role in cancer diagnostics, management as well as therapeutics. Blood, being the fluid connective tissue in the human body, carries a lot of things in circulation which can be used as biomarkers to reveal mystery behind several diseases and aid in their management. In the recent past, blood-based biopsies or peripheral blood, commonly known as liquid biopsies, have received great importance with respect to cancer diagnosis, prognosis, and monitoring progression. The image shows a tumor cell detaching from the primary tumor and traveling in the blood vessel along with other biomolecules and cells in circulation, which can be collected in a single blood tube upon a simple blood draw and can be isolated for further analysis to collect information in order to make a diagnosis or design a therapeutic. Circulating tumor cells or CTCs or any other cell can be identified using fluorescence staining method. The first method is direct immunofluorescence, where the CTC antigen is detected using a specific primary antibody with fluorophore attached to it, whereas the second method, known as indirect immunofluorescence, involves binding of unconjugated primary antibody specific to the CTC antigen, followed by binding of fluorophore attached secondary antibody to the previously attached primary antibody. Once the cells are stained, they are viewed or imaged using a microscope that has filters to detect fluorophore tagged cells. When a light of certain wavelength called excitation light falls on the fluorophore, the electrons, upon absorbing the light, get excited, leaves the ground state to enter the excited state. The electrons in the excited state, after staying there for some time, starts releasing a lower energy light of longer wavelength on making their way back towards the ground state, and this released energy called emission light is what we view or image through the microscope. Another technology for mutation detection, especially the rare mutations, can use a tube of blood from which plasma or serum can be separated for isolation of circulating free nucleic acids, which are then subjected to droplet generator, which divides the isolated nucleic acids into separate oil emulsion droplets, and all these nucleic acid droplets undergoes polymerase chain reaction to detect mutation of interest. Another way of deriving useful information using a blood tube is to separate plasma or serum and subject it to sequencing to generate a profile of DNA sequences, which involves clustering of target nucleotides using fluorophore tagged complementary nucleotide, generating a plot showing the order of nucleotides and frequency of rare patterns. Subjecting CTC to single cell sequencing can also generate very useful information pertinent to mutations and thereby designing therapeutics. Exosomes are extracellular vesicles secreted by almost all cells. Exosomes secreted by cancer cells have recently gained importance with respect to cancer diagnosis and therapeutics. They are suspected to play an important role in blood vessel formation to support cancer tissue enlargement, cancer cell migration and invasion, escaping immune surveillance, and metastatic niche formation, reaching the potential metastatic site before the arrival of circulating tumor cells. Exosomes can have a wide range of applications under diagnostics, therapeutics, and as delivery medium. Under diagnostics, antibody-based screening for general exosome marker and cancer cell-derived exosome-specific marker can be performed. In terms of therapeutics, antibody-based cancer suppression can be achieved by modulating the cancer cell exosomes to block the communication between the exosome and cancer cell that could hinder the tumor progression. As delivery vehicles, exosomes can be used to deliver drug to target organs in the body, which could be the safest way considering the issues surrounding currently available viral as well as non-viral delivery vectors. Some recent studies showing platelets able to confront with tumor cells in the body via biomolecular signaling and capable of ingesting circulating mRNA make tumor-educated platelets or TEPs a potential candidate for cancer management, and generating their mRNA profile with the help of RNA sequencing can lead to identification of tumor type associated profile. Thank you for watching. If you like the video, please hit the thumbs up icon and subscribe to my channel, The Curious Biotechnologist. Stay tuned for more videos. Stay curious and happy learning.